Hello, g'day, good evening. Thank you for listening. This is a medical cannabis show and I respect the law and I hope you do too. The information provided today is for educational and scientific purposes, so the information is fact-checked. Any of the papers that you'll see I'll go through, I'll put in the description below for you to peruse later on. And I don't know what the topic is today, so I'm hoping that the viewer's going to have some cool discussion or better still questions, and we can elaborate on some of those topics. So I like to focus on plant science, soil science, and microbiology because that's what I study at university. And I've got 30 years plus experience, so it eventuates to something pretty cool. And I'll show some slides. So if you're watching this back, you might want to skip through until you see slides or the screen change. And that's usually at 5 or 10 minutes approximately. And if it's only gone for 10 minutes, that means there was no topic today. So I'll wait for till 2 minutes and then get started. So another 40 seconds and wait for the cool folk to arrive, like Genetic Memory Farms and Shipwreck. Oh, and sure, bro. G'day, g'day, g'day. Yes, I'd like to speak to you, sure, bro, actually. Check your Zoom messages, please. And, so I've got another 15 seconds and I've got to think of something to say. Or hopefully you can write your questions. If you've heard any like weird myths or anything a bit whack or something that you just want clarification on, even if it's really, really simple and what other people think silly, well, I don't think it's silly. So just whack it in chat and we'll see what eventuates. I'll try and elaborate on the topic and share some of my slides and some experience to elaborate. Now it's after 2 minutes. There's been a little bit coming through. So, Genetic Memory Farms. G'day, g'day. Hello, Shipwreck. And sure, bro. Nice to see you fellas. Genetic Memory Farms says, so I checked a breeder with Cherry F3 and he says this 70 to 75 day, not that long for an African. Yes, I am wondering if it is just ripening from the bottom up with the top colas finishing last. Oh, yes, you could get some different expressions. I've heard of that. I've heard of people topping it and then waiting for the bottoms to freshen up later on. You can, yeah, because there's a big difference between plant maturity and flower maturity. So you can see each cola or each lateral that goes out, it might finish different. And you've got a cultivar that does. I'll just keep reading down. Blinky, hello, Blinky. Nice to see you, Blinky. Thank you for support. Chiro. Very good stuff. Yeah, so back to the only thing that has come up. Put your questions in if you can, please. Talking about plant maturity. So, actually, there's another person asked about that too. Let's have a bit of a squeeze. Some plant maturity stuff. So I'm just going to share the screen now and get into some plant science on secondary metabolites and those plant maturity. So it would be to do with trichomes and cannabinoids. But we're going to assess the trichomes first before we look into the trichome and see all the different properties. So there's a fair bit here, as you can see. I'm going to get to the better ones. in maturation size. Here's a good paper that goes through the trichome maturation, how it matures. Right, it's a bit in detail. So the diameter of the largest droplets. So they're saying they're actually about 20 micron across if you put it under the microscope. That's the average. You can get them way bigger. I've seen ones at 200. The extractor's dream. So what's this really showing? How can I help you with this question? The leaf sessile. It matures first on the leaf. So if you're starting to get trichs that are changing their color and going from clear to milky to amber or reddish, that's the maturation stage. But the leaves mature first. So you want to look on the flowers, on the perigonal bracts, the calyxes. And, oh, cool. This says, here you go, the leaf, the sessile ones. So sessile ones are just without the stalk capitate. So that's these pictures up here. And then the stalked ones are the mature ones, and they've got the stalk on them. So you can see here the largest droplets in diameter. They reckon they get smaller ones on the leaves, and then the sessile ones are next biggest, and then the largest ones that are mature are the stalked. And what else is interesting in here? This is a good example of them. So this is the sessile ones in the middle, and this is the stalked capitate. You can see it's elongated cells underneath. This is pretty cool. It's electron scanning microscope one, this one. And that's one that's starting off. So it will be in the middle. It could be finishing like that, but generally that's in the middle. But who has an electron scanning microscope? We just have these normal microscopes, which look like this at the best. So pull them up, you can see little sessile ones, and down here that are indicating that they're finished. So you can see the different stages here. So this is clear. This is a bit milky. And down here is fully finished as it's ripening. And it ages faster if it's at this state, so it's best to be around this, but it's all on preference, because as the different properties inside of the head there age, they will change. So it depends on what your body makeup is, to if you like the ones that haven't changed quite much, or if you like the ones that have changed a lot. So you can do a little test on yourself. This is what you expect to see under the naked eye. And then you could get your phone and you can zoom in, so you can do a bit. And this is on the bract, you can see for sure. It's not on any part of the leaf. Oh, look, there's a uni up here. Unicellular, no, none. That's got cells in the middle, so it's still got little bits in the middle of it, but it just hasn't done. It's not big and bulbous like these ones. But yeah, this is well and truly done. If you were to zoom out, so that flower has reached its maturity. Don't know about overall plant, because you can't see if the rest of the plant look like that. Yes, the whole plant's reached its maturity, but you take it on its phenotype. That's some cool looking funky different purple expression. That's probably after you'd be looking at it after you've taken all the tops off. Yep, in a dry sift. That's what it looked like. I'll go back the other way. Let's see what else is here. There you go. There's its changing of its cell structure inside of it as it matures. It's different terpenes inside. Go from monos to sesquas and sesquas. So it's the terpenol, the isoprene level, 5 and 10, and that's up to 20, I think. Let's look at some other cool ones. Look at that nice looking round thing. Isn't that? That's actually real. That's a scanning electron microscope, but that's actually blimmin' real. Isn't that cool how just perfect it is? I don't know. It's the surface of an anther on the male flower. So that, here you go. This is just finding a little bit ones of the definitions of them. If you want to get into it, some terms used to describe trichomes on their surface, the different types of... Actually, I'll stop sharing. I'm getting a bit excited here, see if there's any questions. That's right, we're still on that. It's on screen. Sorry, everybody. I did check that, yeah. Okay, GMF said, a flower under a blue spectrum. Do you think this could alter the ripening takes place with bluer or redder light, slow clouding of trichomes, and keep clearer heads versus amber? The blue spectrum, yes, it does. It helps with photosynthesis. It photosynthesizes the yellow carotenoid pigments, and that's why some old school people use the metal halides in late flower. So I hope that answers that question. So yes, it's going to photosynthesize. You're also going to get the best out of the more genetic potential out by doing that, because the energy that's produced from a spectrum, a yellowy spectrum, will not produce as much energy as the blue spectrum regarding carotenoids. So it's going to metabolize a bit better. So that's all you're just going to produce, and it could ripen faster. Well, I'm not sure, because it's photosynthesizing. It's just doing its job. So it's reaching its metabolism potential. So it can produce a lot of energy, therefore go through its growth processes. I'd say it's a genetic thing or a gassy thing if you've got something that's finishing earlier. The next thing says, will bluer or redder light slow clouding of trikes and keep clearer ones, or the amber? So will blue or red light slow the trichomes? Well, it might age it faster. So it might, yes. So if you have, for instance, a light that it doesn't agree with, it might think, oh gosh, this isn't good. I'm going to go into reproducing, and it might see it as a stress and go into the stress phase, which is giving more secondary metabolites. That's a pretty big topic. I've got some, I'll see what the other questions are, and I'll go into that if there's no other questions, because different spectrums on photosynthesis do have a effect. Just scroll down here. Blinky says, I have a question about breeding. I need help. Oh, with first time in 30 years, Rusty, can you crossbreed an auto with a fem easy? Can you crossbreed? You can grab the autos. Aren't very good to mess with because they don't like it. The ruderalis just doesn't enjoy being messed with much, but you can grab the pollen off it. So that's a big difference. So the question was, can you crossbreed an auto with a fem easy? Well, yes. You can put a silver blocker on it, on the auto, and that's going to block its ethylene and start and produce the male bits, the pollen sacs and the anthers that you want. And they have the little pollen grains inside of them that, sorry, it's not anthers. Anthers is the female. Yeah, I'm going to show some taxonomy just to pull that, fix that up, what I said, because it's not right. But in short, yes, you can grab taxonomy. Grab the pollen and put it on the one you want to pregnate. So taxonomy for the, I know where it is. Taxonomy, come on. There it is. Am I still sharing? No. Okay. So this is just to clarify the parts. So up the top, you've got the male bits. So the anthers are inside of the sepal, and the sepal's the round bit. It just looks like it's the balls that people used to say. And they do call the anthers bananas because you can see they fair dinkum, even under a microscope, they look like a banana. It's cool as. And in the female bits, the bracts, they got the pistils coming out, which is the stigma up the top and the style down the bottom and then over in the middle. And the pollen grain comes out of here, which has thousands inside of the anther. So once the anthers come out, there's also some inside of the sepal, but mainly when you cut that across there, it'll stacks, it'll just explode. If it's under pressure and mature, it will explode with pressure, and that's what lands on the top here and travels all the way down the stigma and the style, goes around, up through the micropyle, and then fertilizes inside here. Depends which cell it hits to which eggs it's going to grow with, because in the middle there's some triploid ones that can reach if you've got like a double fertilization process. But in general, that's the taxonomy. So there's the female bits, the bracts, and the stigma and style, and then the anthers are on the male bits. And now what the question was, can you crossbreed with an auto? So yes, so you'll go and put some silver blocker on it, some sort of silver nitrate or goidal silver or silver thiode disulfate, SDS, and you'll spray it like that. And if you really want to know about it, I'll put a video out on a few on breeding. So go and watch that, and it'll elaborate a lot more, and then you can ask some questions to fine-tune it if you'd like. I hope that helps. Will it cross? Yeah. Hey, Sunshine. How you going, Sunshine? Nice to see you. G'day. 420 Fred. There you go, mate. No. Someone's complaining in chat that the bananas, no, don't want bananas. In other words, don't want it to herm out. So your plant, it goes through a stress phase, and then it'll start to reproduce itself, and it'll throw out those anthers or bananas. But you can work with that. You can pluck them off, or you can let them go, or you can move things to a place where it's not going to affect other ones, like out of the draft, out of the wind. And to be honest, it'll have to be a pretty decent stress event to make it induce and ruin everything. Otherwise, it's generally not a problem. It's, yeah, you can work with it. And you might get some backups of some good ones too. And it's always handy to have backups, because if you go through diseases or viruses or something like that, you want to have backups of what your ones that you like to grow. So, oh yeah, Blinky, thanks heaps. Oh, good. You're welcome. Yeah, check out that video. It's very handy for getting into that. Back with the, is there any other questions? Whack it in chat, please. The blue and red light. Let's investigate that more. How blue and red light affects photosynthesis. What do you reckon? Reading. No, plant science. Might even be a photosynthesis. Yes. And circadian rhythm. Look how many photosynthesis folders there are for the difference. But I'm going to try, I'm not going to go into great detail because you can see there's heaps of different detail. I think I did a course just on plant photosynthesis. All right, let's just go through the highlights. We'll keep that one for later. Processes of the plant. No, we're not going to get into it that much because I don't want to bore you or explain things that are going to bore you. But this is a pretty rad graph. This shows the wavelength down the bottom on x-axis. Y is the absorption amount. So it goes from 0 up to 100, and you can assume it's percent. So down here, it's got the wavelength, which 700 is up near the near far red, so that's up 700 odd, and down here the blue is in the 400s, and then you go with your X-rays, your UVA, B, that sort of stuff down below, and then the green's in the middle where it doesn't use much. So you can see from chlorophyll A, there's a few different systems that the photosynthesis goes through, chlorophyll A and chlorophyll B, and you can see that's its action absorption spectra. So if you're looking at chlorophyll A, that's the peak you're going to get through different wavelengths. Chlorophyll B is up a bit higher, and then it comes and reaches up here. So it's just in general is the black. And then the carotones there. So see, with your carotones, you've got that's the carotenoids over here. And you can see that the spectrum that it loves is the 4 to 500, which is the blue GMF. So that's why you're getting a better response out of the blue in regards to the energy produced through photosynthesis. And up here later, I'll show the actual amount of kilojoules produced difference that you can see. So if you want to work out the maths, it's quite an amount too. Oxygen-dependent bacteria accumulate near illuminated with blue and red light. Okay, that's more so to do with bacteria because they're autotrophs and they want to get the most photosynthesis out of it. They use a different pathway as well. They got extra pathway. I'm talking about plants. Chloroplasts, sites of the photosynthesis. So there's the different where the chlorophyll in the thylakoid membranes are in the plant cells, and then wherever the green bits are, and these are cool under the microscope. You can see them dancing around. If you put like a brighter light on, they'll react faster, and lower light, they go slower. So that's indication of their energy that they're producing. This is a really cool one because it shows the absorption ratio of the green leaves. So the greens bounce back, so I can tell the different things that's actually getting absorbed. And then it comes up near the infrared, and not many of the spectrums go above sort of 700 because that's the range which the human eye sort of sees. This goes a bit higher, and you can see that there still is an amount. Look, we're up here on 10%, so it still is absorbing 10% as it goes up. And look, it actually goes higher above that into the infrared range. Isn't that interesting? And here's the difference energy that it produces. We were talking about before about the amount of kilojoules per mole that it produces through blue light. You get 274 and red's 181. So you get a lot more energy produced under the blue light to go down and photosynthesize and produce ATP, which is the cell currency, gives us its energy. Chlorophyll and photosynthesis, the different pigments. We were talking about chlorophyll A before. That's where it's ranged, and chlorophyll B is higher. And carotenoids and anthocyanins, as they go up a little higher again. Sort of covered that. Don't really want to get into photosystem 1 and photosystem 2. The photosystem 1 is in the range above 700 nanometers. So it really interacts with dull light or around sunset and that types of thing. Photosystem 2 is the main one. That gets pretty technical, but that'll do. The energy can be produced at night. So see, our light comes through with photosynthesis, and you get light reactions that react in the chlorophyll, and they produce the ATP down the bottom. But what happens when it goes dark? Well, the plant doesn't sleep. It still produces energy through the Kelvin-Benton cycle, and it uses its starch and sugars to do the conversion. With rubisco, the enzyme breaks down, and it still can produce the energy that it wants. And a long story short, transpiration. No, we're talking about photosynthesis. Oh, and this is brilliant. I've never seen this ever anywhere. Well, look at the range that throws photosynthesis in right up to 3,000. Most go up to 750, 700 nanometers. The far red, which is our color spectrum, you can see here. But look at the reactions as it goes higher. Isn't that amazing? That's why the sun is very hard to be reproduced inside greenhouses and things. And as it goes up, you've got different energies that are pulled out at the different wavelengths. And the dips are the gases that has been formed, which it doesn't get out, it just gets water out. But yeah, it's just a solar energy distribution in the atmosphere. That's pretty cool. I don't want to go into that much detail. I think this is another, the red and blue light, most effective in photosynthesis. So the rate, so this is like just a colored version of the action absorption ratio. So you can see how much energy it gets out of it. And that's why a little bit less kilojoules, I was saying before. And the red and the blue. Appropriate UV treatment increases plant yield. It increases the stresses because the UVA and the UVB, UVC is mostly burnt up in the atmosphere, but there's UVA isn't very good for the plant and it stresses out, so it produces secondary metabolites, which we all know are pretty cool. This chlorophyll, this is done by algae and microbes. No, let's find other interesting things. Seen enough of those wavelengths. There's some good studies on circadian rhythm. How does the plant actually know when to do things? It actually does have a circadian rhythm. It's fascinating. Circadian rhythm, no. Phytochrome response are at 3 stages. So there's, no, we're not going to go into that then. We don't want to go into that much level. They're the actual names of the red. That was the name of the red pigment. Factors affecting photosynthesis is light intensity, CO2 concentration, and temperature. So any one of these things can limit the photosynthesis. So if you've got all these things in check to what the plant requires at its potential growth stage that it is at, it will be ticking. If you can get all these numbers up in the high 90s to 100, that's going to give it maximum metabolization. The rate of photosynthesis becomes constant. Either the concentration of carbon dioxide or the temperature needs to be raised to increase the photosynthesis any further. So x-axis is the light intensity, y-axis is the rate of photosynthesis. So as the light gets brighter, we're talking about a mature plant here, so as the light gets brighter, it's going to increase its photosynthesis amount until a point which it's reached, which equals the amount of carbon dioxide. So if you were to put a bit higher amount of carbon dioxide in, that's going to allow the plant to create more reactions, and therefore the rate of photosynthesis goes up a bit higher again. That means, this is getting a bit more technical, but it's very, very particular, because when plant goes through its senescence, it shows anthocyanins. I've made a video on how you can get more anthocyanins produced in your cultivars, and the anthocyanins, it's got plenty of antis in it, antioxidants, just brilliant, good health. So it's important. But look at the wavelength. Oh, what's that? It's in the green, you say. Yes, it is. It's at 550 odd in the green. And they said green doesn't work. Well, it does if you have anthocyanins. So it won't be photosynthesizing or producing energy or for any of the other pigments, but the green reacting with the purpley pigment does. And you can see on the left here, it's quite high. That's, what's that, 35 odd? So if chlorophyll A in the red goes up to 79 or 80, this is nearly half of that. So that's still definitely to be considered with the spectrums of the plants. We've seen enough of those ones. The changes in light intensity. Oh, I won't go through that. You can see there's some blue lights there put in through aged to maximize the yellow carotenoid cells. No. Here you go. This is fantastic elaboration of light reactions on plant leaves. So here's the light treatments. So you can, I'll just go top axis. Light treatments are in here, the different species, roses, lettuces there, the responses and what happens, and this is the references to the papers that you can put in. So for instance, down here, blue LED lighting in grow chambers with lettuce. So increase the concentration of compounds compared with triphosphor fluorescent lamps. And that's the study that you want to put in. So these different pigments for different situations work well. I can vouch for the blue. It's, yes, got some extremely good reactions. And this is to go on just with the different other pigments. Yeah, easiest way to say it. Anthocyanins, antimicrobial activity, and protect cells from excess light by absorbing blue or UV light. Involved in flower and fruit coloring and serves as an insect attractant. Cool stuff. I see the chat going off. I shall just put that down. And you'll see everything. Holy mackerel, I haven't been in the chat for a bit. All right, I'll stop this. I'll come back to those different examples. And I'll just read through chat for a smidgen. All right, I'm up there. I have a few questions. All right. Just trying to catch up a bit. Nose, thanks. G'day, g'day. Yeah, he's Raptor. How you going, Raptor? I see you, mate. Another way. Yes, it is. Yes, maximize your genetic potential with photosynthesis. Well said, Raptor. So we'll go back to sharing the screen, if you can say it right. So what's next? So out of the light that reaches the surface of the Earth, UV is only 5 to 6%, and most of the UV is burnt up in the top atmosphere in the high reaches of the atmosphere in the ozone layer, to be precise. UVB gets a little bit through, and UVA mostly comes through to the surface of the Earth. Relatively, what's that? On the y-axis, relative proton flux and the wavelengths. Okay, so that's just the different colors broken down in their respective wavelengths and their amount of energy on the side. Pretty cool 3D graph, eh? You can see, all right, cool white LEDs, you're going to get that spectrum, so pretty broad. Red LEDs, you're just going to get that particular spectrum, which are used extremely good in different areas. A warm white gives you more of the red, and green, well, it's the no photosynthesis. No, sort of covered. We've covered that. There's a way to break photosynthesis or to beat it. That's more so photoperiodism. Photosynthesis. This is photoperiodism. While we're on the topic, it was always thought you're supposed to do 12-12. If you do 14 on, 10 off, you're going to get stretchy, leggy things. If you do 10 on, 14 off, you're going to get things that are pretty chunky. I was quite amazed at that. So in a CO2-deprived environment, this might be a winning move for one to take. All right, is that it? Yeah, you can see the difference. Quite. All right, that'll do. Did that help, GMF? I hope it did, mate, because I can go into it a hang of a lot more if you want. I suppose there's not really any messages coming through. Do you want me to elaborate a lot more on photosynthesis? What do you reckon? It takes 30 seconds for the slides, for the delay to come through before I see it. So I'll just wait a little bit and see if you just want me to elaborate that, or maybe there's something else can be put up and I can go into your questions if you're having any things of concern. All right, well, if I don't see anything in a couple of seconds, I'm going to go back into photosynthesis, I think, because I don't know what else to talk about. Could always leave it early, but I'd like to do what the viewers want, so it's up to you guys and girls. All right, I don't see much direction coming through. So that's a pretty good one for senescence from changing its pigment. Sunlight and traditional light source versus spectra. Yeah, you can see why it's very hard to be reproduced. It's just brilliant all the way through. Incandescent light, they try hard. HPS, yes, it gives the blues and the reds, but what about the greens and all those other different spectrums, especially beyond in the infrareds? Fluorescent light, that was always pretty good. Basic functions. Photosynthesis produces the energy for the plant to do its metabolism, to complete its metabolism, which it breathes in and breathes out. It respires and transpires. So it respires in mostly through, well, I think it's all through oxygen through it, and it's only through its roots it gets its oxygen source. And it transpires out through the cells up the top, which mostly is the stomata. There's one stoma or heaps of stomata, and they grow, it depends on the growth conditions that's been put under. You'd like to see, well, in bigger ones, even like we're talking of the polyploids, they grow bigger again because the cells are bigger, so they get larger. That's a good way to tell if your cultivar is a polyploid, a triploid, or a tetraploid. What else can be said here? That's about it. Yeah, it goes, respires in, like we breathe in, and we transpire out, we breathe out. So oxygen comes in and through there. And I don't want to repeat myself. That's it. That's it on the highlights for that one. Shipwreck says, I say keep going. This is all really good to know. All right, mate, I'll try and get they were the highlights in them, and then I go into break off to the individual ones. So, well, photo-respiration is pretty bad. Let's talk about that. Unnecessary evil. Oh, it's when the, oh, I'll just go and have something up here to explain it to. Oh, photosynthetic equation. This is something that's easy. It's not a C3, we're only C4 plants. CO2 is not entering the leaf since the stomata is closed. Thus, the CO2 is being used up in the Kelvin cycle, which is the night cycle that it uses, and not being replenished. Concentration of CO2 decreases to less than 50 ppm, and it's dangerous for the plant. So oxygen ratio in the leaf will increase relative to CO2 in concentration, and then there's an oxygen problem. So the enzyme Rubisco will stop using oxygen and start using CO2 inside of the plant, and it's bad. So photorespiration. Discoveries, no. Oxygen. As the concentration of oxygen increases, the rate of photosynthesis decreases. This occurrence is due to the phenomenon of photorespiration. As the concentration of oxygen increases, the rate of photosynthesis decreases. Okay. Greater inhibition of photosynthesis. No. Even the normal ambient concentrations of oxygen, which is 20.9%, in itself are sufficient to slow down the rate of photosynthesis. Yeah, remember that, actually you don't remember, but Oh, this is the one I was just going to quote. How brilliant. So oxygen concentrations are double from normal at 21%, but oxygen concentrations reduced to 2% equal that photosynthesis doubled. That didn't come out too good. So if the oxygen concentration is extremely low, 2%, which we probably can't survive in, photosynthesis is doubled. So in other words, the plant likes a low oxygen concentration. So if the oxygen concentration is normal of 20.9%, there's a decline in the rate of photosynthesis because it's too much. It's, well, it likes it lower. Curiously, this post-illumination burst become more intense if the oxygen concentration in the atmosphere was higher. So this is a post-illumination burst of CO2 showed in a photosynthesizing leaf. So x-axis is the time after the light's turned off, and the y-axis is the rate of CO2 production. So lights go off, and you see a spike in the CO2 production, which is normal. And it comes back down, and it'll stabilize. So see that oxygen is a lot higher. So it shows a higher amount of CO2 production. And here it's in a lower oxygen state of 2%. So it's respiring lower. So what's this? It's pretty similar, but it's just giving you the difference. Okay. So it's really marginal by the photosynthesis. But if you want to fiddle around with genetic potentials and things like that, you might find that a lower oxygen environment will benefit it. So the burst of CO2 was directly proportional to the CO2 concentration. Okay, very good. I'll skip through this because there's a fair bit in here. Photorespiration occurs in plants when relatively high CO2 compensation point, 50 ppm or more, like I mentioned C3, this is C4 plants. Photorespiration, the uptake of oxygen and the release of carbon dioxide in the presence of light without any ATP production. So conditions of photorespiration is when the temperatures of the canopy get too high. So when the light intensity is usually too high, you find canopy temperatures too high, and it can go up above 37 Celsius or about 100 F. It actually depends on your cultivar, but that's from what I've seen. But I like to play around with maximums of things, push amounts. But that's when you'll generally get it. So you might be thinking, oh, cool, it's really hot in here. Yeah, they're going to love it. No, they're not. They go into photorespiration. It's too hot, and they can't do their cell metabolization process. And light intensity is also a bad thing. So if it goes too high, they can't do anything with it. It's like you getting thrown out in a hot summer's day when it's in Arizona desert with no shoes on, going, right, you'll be right. You won't blooming be right. So conditions of photorespiration are high light intensity, high temperature, high O2, or low CO2. So that's something to keep in mind, low CO2. So if you've got low CO2 below 400 ppm with things sealed up, you can be entering photorespiration and not allowing the plant to do its proper growth patterns. Well, I had to do an assignment on Rubisco, and it is pretty Rubisco. Most enzymes, they only work breaking down one thing, but Rubisco does two things. It works on oxygenase and carboxylase, two enzymes. It's like a freak, but it's pretty cool. It goes into Rubisco a fair bit. Okay. No. 102 is too high. No, this is getting in technical bits that you're not going to enjoy. 3 organelles involved. Oh, that goes Mechanism of photorespiration. Looks like it's all covered. Is photorespiration entirely wasteful? Not really. It's, considered a short circuit. It's not producing as much energy as it could, but it's still functioning. So there's no ATP or sugars generated. It drains almost 50% of fixed carbon on a hot sunny day and evolutionary baggage to carry. So it's a way that the plant functions when things are too extreme. You can call it perhaps a safety valve. Acts as an electron sink for reactive oxygen species formed during light reactions. And you know the reactive oxygen species, they're bad. Peroxidate, they're the bad things that really harm. So you'd need antioxidants to break them down to function well. Same thing with human bodies. That's why anthocyanins are fantastic pigments. Photorespiratory mutants have proved to be lethal under certain conditions. So yeah, you can change things around. You all know about breeding and stuff like that. Very good. C4 plants perform the Kelvin cycle in a different location from the C3s. Righto. I only want to talk about C4s. That's the end of that cycle. I am sharing. Yep, good. What else? I want to keep things interesting too. What else is down here? I'd say keep going. Oh, yeah. Sorry, it's sunshine. Oh, okay. What other questions? Got 13 minutes to go till an hour. So what question, how can I more so elaborate on photosynthesis and photorespiration? There you go. I think I'll just try and do that because there's no questions. If you've got your questions, whack them in because otherwise I'm going to try and elaborate. Hang on, there's one to come through. Here we go. GMF says, so could this high temperature, low O2 environment be something to what happens in a Malawi cob? A Malawi cob is a ground cure. So they do curing in low oxygen environments. That's what a Malawi cob is. It's like a ground cure. There's a few ways to do it. So I'll finish reading. GMF says, I will cut the plant and thinking to ferment this sealed at 40C at 24 hours, then 30C for another 96. AussyCC, what do you think is happening in this cob or ferment? So in a low oxygen environment, things happen differently because microbes aren't using oxygen as a electron donor anymore. They're using nitrate or nitrite, and then it goes down to magnesium oxide, and they use anything that's got an oxygen electron attached to it as a energy source. So that's how the breakdown process works with the microbes. So back to the question. So could this high temperature, low O2 oxygen environment be something to what happens with the Malawi cobs? So with ground fermentations, is that how it works? Is that what the question's asking? You're a good fermenter. You know about that low oxygen environment quite well with your trade. What do you think, mate? I don't know how to elaborate any more with that. Oh, the GMF says to go on to say he will cut down the plant and thinking to ferment it for 24 hours and then 30C for 96. So the first fermentation at 40C for 24 hours and then do a 3-dayer You played around with more ground fermentations than me with the Malawi cobs. Actually, you've done a fair few Malawi cobs. I've done that ground, the ground way that they do in Asia. So I'm just a bit sucky and trying to help you out a bit. So he says, I will cut and thinking to ferment it in a sealed at 40C for 24 hours. Well, what's that going to do? 40C, it's not enough for pasteurization to kill microbes at 67, or I think it is the minimum. So why the elevated temperature? All it would do would, the temperature would be like nitrogen assimilation happens with temperature. So you're going to get maximum nitrogen breakdown at that temperature. So the microbes will be more active at that temperature. So this process might start faster. And then 30C. So if you're going to keep your microbes working at 30C after that, they will only do their processes. Mesophilic microbes range their best at about 35 to 40 Celsius. And then it tapers off either side of the graph into the next ranges of microbes. So I would think that, yeah, I'm not sure what the outcome is, but it sounds pretty good. That's all I know about the topic, if that helps, mate, with your experiment. I love experiments because even if the experiment doesn't work the way you want, you've still gained information. And that's brilliant because you can use that to adapt or modify and possibly get the outcome that you really want. So every experiment's rad. So I support the people that do that. Good on you, mate. Keep up your good experimenting work. And he shares his results too, which is fantastic. Do you think? Hey, there's Lene. Hello, Lene. Sorry, Mr. Show. RSO did help me in heavily yesterday. LOL. Very good. Maximizing the medicine. Brilliant. Nice work. Cheers. Yep. Oh, Raptor says, do enzymes play a role in trichomaturation? Oh, that's actually, I've just done 5 lectures this week on enzymes, plant enzymes. Can you believe? Is it in their signaling cascade pathway? So the end result, well, do enzymes play the role in plant or in flower maturation? Well, it's a part of senescence, and if it goes through a stress phase, yes, because it'll be going down releasing the bad stress gases, the abscisic acid, or more ethylene, so it won't be going into its growth and development pathway. So if those enzymes are triggered, yes, depends which pathway it goes down. But if the good enzymes, not like, for instance, nitrogenase, that's what breaks down nitrogen. So if your plant's limited as it goes through its growth phase, you'll get that happening. So do enzymes play a role in flower maturation? Yes, is the short term, because it's involved in their pathways, and it's how you trigger different enzymes to different pathways you get. Like if you come up to a set of traffic lights, there'll be some nitrogenase sitting there, and you need a nitrogen molecule to react with it, a car going through, and bang, it breaks it down. So you need these things to be linked up. But if you don't have any one of the two, the reaction can't take place. I hope that helps, mate. I didn't really elaborate much. Hope that helps, Raptor. Let me know if it doesn't, because that was a very short answer, considering I just did 5 half-an-hour lectures of it a few days ago. GMF says, I am trying to guess what is happening. We use 40C to ramp up the lactobacillus ferments for food and beer to increase acidity. I think the second stage ferment just stabilizes it. The second stage. The first stage produces the energy, the catalytic reaction, which produces all of the, and then Acetobacter, it metabolizes all that, that's been produced. So if you put heaps of sugar in, for instance, it'll produce like heaps of way more sugar. It'll produce way too much, and you'll get too much gas from Acetobacter. I hope that helps. I think the second stage ferment just stabilizes it. The first stage, the lactobacillus produces it all that the next microbes can use. So the yeast needs to expand and produce the gas, so it needs certain elements for it to function, and the Acetobacter also needs certain elements for it to function. Yeah, that's what I reckon. Hello. GMF says, my guess is the presence of Lactobacillus plantarum on the plant, just like on cabbage. Lactobacillus is not very good as a microbe to focus on. I know Korean natural farming does it a lot, but it produces genus produce heavy biofilms. That's why they're good in preservatives, because they don't allow other microbes to proliferate or to even get a start, because they'll keep pushing them out from their biofilms that they expel other microbes. So yeah, I've never gotten into the lacto genus for that reason, because I want to build a big diversity of cultures where all my microbes do certain things in certain areas. So I'd like them all to be present, but in certain areas turned on. So when the plant's in flower, I might want more microbes that produce gibberellic acid, or when it's stressing, I might want more microbes that produce fungicide, as an example. I love the questions, though. This is really terrific questions. Keep them coming, please. Or keep all your statements and stuff. Keep them coming. I love ticking my brain over. Fantastic. Raptor says, yes, but I'm now curious if an external application of enzymes on the plant or soil has an effect. Well, yes, but it has to be the right one, because cellulase, for instance, isn't produced by many microbes. Bacillus pumilus produces cellulase. But if you've got, all right, talk about bacillus. If you've got a compost heap and you want to break it down pretty decently, you want it to cycle and produce to reduce all of the properties of that. And that's all of the lignin, the cellulose, hemicellulose, and pectin that's made up in cell walls of plants. So they're the elements that you'll see inside of it. So you want pectinase, a microbe to produce that. You want a cellulase, microbes to produce that, and then you'll get the reactions happening faster. And then inside of the plant, you want those reactions to still continue. So the plant should, if it's in good health and not gene deprived or a mutant, it should have its 30,000 pairs of genes in place to fix its immune problems and things like that. It's the soil, I suppose, or your substrates that you've got to worry more about, because microbes can manipulate them. It's not the plant itself that it can't take care of itself as well, because its endophytes aren't around the soil. They're inside of the rhizosphere, not the rhizoplane around the soil roots. Back to the question. I'm not curious. If an external application of enzymes on the plant or soil has an effect, well, on the soil, it breaks down all the bad things and it has a really good effect. It really does. It can even balance things. It depends if your substrate is a healthy one or a non-healthy one. If it's not healthy, it would very much benefit it, because it's going to break down all of the heavy metals that are also inside of the tissues and the vacuoles that are in the plant cells. So you don't want those. You want them to leach out, or them to be chelated with other elements that aren't going to be taken up by the plant's roots and can be possibly swept away or absorbed by fungi or other things that's not in your plant. And would an external application that handles the substrate And will an external application of enzymes help on the plant? Oh, yeah. Well, external. Well, a foliar spray only takes up about 2% of the mineral nutrition of the plant. So if you was to spray it there, you're going to help it, but it's only going to be very minimal, I suppose, because it's the amount that it can be actually absorbed into it. And especially if you haven't, there's a few restrictions about the absorption too. If you've got waxy layer on your leaves, the waxy cuticle, it's going to be harder to get through and absorb for external applications. But in general, yes, they do, because enzymes are part of the signaling pathway in plants. So if you can help them out with steps in that pathway, they don't have to work as hard or get the right key to fit into the lock. They've just got it already sitting there. I hope that helps, mate. But there's heaps of enzymes too. There's just not one or two enzymes. There's millions of enzymes. So it's also the right enzymes that you need is another pretty cool question. I hope that helps again, mate. Raptor. GMF says, I think this ferment or cure is just way to ensure that Lactobacillus plantarum is the dominant microbe. That wrapped up. Point. Okay, so I think fermental cure is just the way to ensure that is the dominant microbe. Yes, because that one might work in the low oxygen environments in the ferment. It might be perfect for that if it's a obligate anaerobe or a faculty aerobe. It should do well. They're the oxygen requirements for the particular microbe. Because I don't know much about the Lactobacillus genus, but it sounds pretty good. GMF says to someone else in the chat. Very good. GMF says exactly why I think it does well at preservation. Ah, very good. We come to a conclusion. Fantastic. Yeah. Yep. So you can probably increase those microbes. You'd go to a enrichment culture for Lactobacillus plantarum and look into that, and that will tell you exactly what conditions, environments it grows under, and there'll be some papers under that too. If you look under the Scholar, under the search engine, it will show different papers that have been performed in the enrichment cultures, and then you can read which ones work with your materials. For instance, you might want to use mannitol as a carbon source, or you could just use plain sugar, or you could go with glucose, different things. So there'll be all different enrichment cultures to what suits your application with your inputs, because you only need 5 things to grow a cell. So if you can get all them right, you're on the way. And then you've got to get the right conditions for it. So once you've met those things, it's no worries, mate. No, that's cool. I didn't have to. I was sharing a screen up here. So I was going to share the screen for something, and I don't know what. It's been over an hour now. Is there anything, put in chat now if anything you'd like me to elaborate on? Otherwise, I'm happy to call it a show today and retitle it. What would be titled today? Photosynthesis. What else can I put in? Plant photosynthesis I should put in, because I like to talk about microbes a lot. Plant photosynthesis. We're going to do a bit of photo-respiration. And talked about, we touched on enzymes, but I don't think much, I could put that into the title. Is it just those two? Fair income, is that it? Oh, and light spectrum. It was photosynthesis, light. What can you say? White color. Something, I don't know, something like that. Not sure. Raptor says, do you think chitinase only needs to be present in the soil with chitin present for the plant's immune response? Would it still work if sprayed on the plants with a pest present? Chitinase is, the cell walls of fungi are made of chitin. So it breaks down cell walls of fungus. So if you've got a I'll get back to his question instead of elaborating. Do you think chitinase only needs to be present in the soil with chitin present for plants' immune response? Microbes produce chitinase. It's like their natural fungicide. That's one of their enzymes they expel to be called a fungicide. It's chitinase. So would it still work if plants were sprayed with chitinase? Well, the chitinase molecule, it needs something to react with. So if it's sprayed, it'll just sit there. So it's not like a virus that's a viroid that has looking for a host, that it's just going to reproduce once it gets something living. The enzyme's just going to sit there and wait for its next pathway, like so I don't think it would work too well if it's sprayed on the exterior of a plant. If you had fungal cells on the exterior of the plant, like botrytis or powdery mildew, and you spray chitinase onto it, I think that will definitely work. I think it'll start within a few days, you'll start to see it going all brown and working really well. But to spray it on just plant cells, I don't think it will react and really do anything. It might slowly absorb through the tissue of the dermal layer, but it wouldn't have an effect because chitinase getting into plants' external cells wouldn't really do much. It might, and what a plant does then, once it picks up different outputs or inputs that it doesn't like that, or heavy metals or things, it stores them in the plant cells because the plants are quite good bioaccumulators. So it'll go into a vacuole or a different other edioblasts or different places in the plant where it can store chemicals that it doesn't agree with or it has no purpose for. Did that help? Oh, he's got something underneath down here. Sweet. Blinky says, my niece just showing up for a tour of my garden. Oh, very good. Have a good weekend too. Raptor says, same with an insect pest on a plant. Well, an insect pest on a plant, you'd want to get to its walls and if you sprayed an enzyme on an insect pest, it'd have to be the one that's going to break down its external layer, like pyrethrum. That reacts with the walls to break it down. I don't know if it's an enzyme or not, because I'm not going to lie, that's stuff that I don't know. But that works on soft-celled insects. So if you were to spray that on it, it just starts to break it down. Same with salts of potassium, fatty salts of potassium acids. But I think we're talking about enzymes. Oh no, chitinase, sorry. Oh, if you were to spray chitinase on an insect pest, what would happen? If that's the way I read your question. It would not do much because the chitinase breaks down chitin, and if there's no chitin around, it's sort of like you spraying water on steel, it's not going to do much, but if you spray water on soil, it'll break it down a bit. That's a pathetic, anyway, that's not a good analogy. But it won't do much, mate. I think you're picking up what I'm laying down. So if you were to spray something that the insect doesn't like, so you want to break down its cell walls, which I don't really know what they're made of, so you'd be spraying that enzyme if you were to do in a similar type of scenario, because the chitinase is an enzyme, because the ASE is a good depiction of it's an enzyme. So nitrogen, nitrogenase, or what's phosphorus, phosphatase, stuff like that. Same with the insect. Oh, Blinky says, I continue to learn so much from your channel. Good people. Love to all. Cheers. Thank you for nice words. Glad you learn. Raptor says, aren't the insects' exoskeleton made of chitin? Oh, okay. If they are, I'm just going to check that. I'm not into entomology much. Insect cell wall or exoskeleton? Cell wall, what do you call it? Yeah, I'll share this. If they are, then I reckon it would. I'll just share while we're doing this little lookup. Insect cell wall structure. No, I want I'll just put in chitin, insect cell, insect exoskeleton. Chitin. This might just have it here. No, is insect skeleton chitin. Chitin is the basic structure of a major constituent of a cell wall of many fungi, insects, and crustaceans. It likely exists in waste from processing of marine food. Oh, okay. So it is. So it's the major constituent. So I think you're onto something, Raptor. I think you're going to have one of these major bad companies chasing you for your work. There you go. So that's the fibers of the cell wall. So if you've got an enzyme to break them down, it's going to react with it and break them down. Short answer, from my experience, yes. Oh, we can leave it in German. Okay, that's just the first one. So what's this? Modeling. Oh, that's pretty full on, isn't it? That's okay. Maybe I'm not on the right area. But the answer is yes. Chitin being the major cell wall constituent of insects, it's going to break it down because it's the enzyme. So it's going to react with it. Wonder what about to break down to lyse or chitinase, sorry. It will work. It just showed it there. Chitin, an insoluble structure. Here we go. I could put this. Here's a study. I'll put in the description. I'll keep it up here. Insect chitinases, molecular biology and potential use as a biopesticide. Look at that, Raptor. You're onto it, and you didn't even know about this paper existed. So it's another tool in your, so I think that you've got access to chitinase. So if you can get that actual enzyme, sweet, you've got it. You don't need any microbes like Bacillus thuringiensis that produces it. Or I'd say Metahizium genus also produces it. Sweet. There's your answer. He's onto something. Let's have a look at some pictures and how it works. It's an insect chitinase and its breakdown. Oh, look, they put them. I usually don't click these and just scroll through the top bit. But you can't see nothing now. I'm not going to click it. But yeah, it works. Well, it has to. It's the cell walls. That's stop sharing. That's some good information. I love it how people help me as well. It's so cool. I continue learning. Yeah, aren't the insect exoskeletons made of cotton? They sure are. The major constituent, it says. And Raptor says, do you think if it would be a benefit to include seed sprout tea that has chitinase present on an IPM regimen? Oh, so if you can get a seed sprout tea, so you want to try and get that cycling through the seeds so it can be resistant. Yeah, I think, well, it's short answer, no, because it's just the enzyme, and you giving that to include on a seed sprout tea on an IPM regimen. Oh, for an IPM regimen, so if you're say once every week to 2 weeks, you're going to include it because of killing bugs and stuff. Yes, but keeping in mind, you're probably you might impact on the good ones as well. So if you've got a large establishment of healthy, beneficial insects, you might be knocking them too. That's where you go. That's where the agricultural folk go with the entomopathogenic ones, which just concentrate on pathogenic. The microbes itself concentrate on pathogens and don't target beneficial ones. I think I'm happy. So by including chitinase, yeah, that's what I reckon, mate, would happen. It wouldn't be good for your beneficial microbes. But if you're in one of those, because I know you're an organic head, but possibly if you're a synthetic person who didn't care and used heaps of pesticides, I think it would be really good, because it's like using natural pesticides each week. If you're into that, which I want to make a disclaimer, I really don't like it. Organics is the go. It's sustainable and it doesn't hurt the environment. And you hit pocket. So that's the way I focus on, not synthetic feeding, chemical feeding. I'm not interested in that. I don't really interested in mineralized feeding. Maybe mineralized foliars, but I'm interested in immobilized feeding. Raptor says, I will experiment with a once-a-month application. Only if you're not trying to build up. Oh, hang on. He says down the bottom here, definitely hit it more when under infestation. Well, it'll just wreck your good microbes, that's all. So if you're really struggling with an infestation, biopesticides is probably like, I remember you have thrips a while ago, so curcuminous and Monterensis, and what's the other one for the soil? Hyperaspis. That's how I remember it. It's how you spell it though. But I'd be going with those ones for the soil instead of trying to knock out ones, because I'm interested in building things up and having my microbes work for me, whereby getting rid of the ones on a broad spectrum, you sort of have to start again. But if you want to go down that route, you're probably going to succeed. You might as well just do a soil drench with some isopropanol, or even You know, diesel fuel lasts 3 weeks in the soil. So there's some different routes you can go if you're interested in that. But I'd be going the bios first, bio-pesticides and microbes that are going to assist, bio inoculants that you can use. Oh, GMF says, chitinase is also present in grass seeds, i.e. it is one of the proteins present in flour. Oh, there you go. Very good. Chitinase is everywhere. So if you want a top dress then, there you go, Raptor. GMF might have helped you out. You might be able to look at some grass seed, collecting that, or accumulating that in your yard, then mow it when you see it. And that might be a good source for you, an organic source. Thank you, GMF. Raptor says, true. Best to use less with more diverse methods. Yeah. Well, I'm into increasing the microbes. I've realized that the microbes work. And if you get the right ones, they work well. I've got most of my microbes I'm working with produce hormones. So you know what hormones do. So they don't only give you fungicide, as a couple do, but they also produce hormones, which, like, if you give your flowers gibberellic acid in flower time, holy mackerel, look out. So instead of going and manipulating and sourcing those types of hormones, you can get in your microbes. So that's just another bonus for that. So I try and build up, I'm looking to build up a good soil food web. So there's many ways to grow. It depends on your conditions, your budget, your amount of labor you can do. You might be too handicapped, for instance. So yeah, I support all ways, but I are into the organic and sustainability ways. That's personally for me, but I do support all ways, especially for handicapped people. GMF says a bunch of food contains chitinase. Oh, heaps of food. Yeah, that's good. It's nice to help out. It's good fun helping, isn't it? That's why I do this show each week, just so I can put my little knowledge into things and get experiments processing and people doing the organic way and maximizing their genetic potential. It's fantastic. Is there any, add some, it's an hour 20 now. Don't get me started on this microbe topic. Some good things coming out of these microbes. I bloomin' tell you, only positivity. Oh, sorry, with the right ones. Yes, I had to add that, because you can't just throw them all in and expect good work. They've all got to function within each other. Remember, there's 6 types. There's competitive, there's parasitic, there's all sorts of ones that don't work with each other, like lactobacillus. But I'm not going to talk about microbes, because today was photosynthesis. And chitinase, I'm going to put in the topic, thanks to Raptor, because there's quite a few papers I'm going to put in the description, or at least one, sorry. So, yeah, can help out people with chitinase interests as well. Well, I think that's it. I want to thank everybody for helping today. Use Folk and Chat, really make the show. I'm just here to steer it in different directions with information. But just bloody excellent. Just good work, chat. Thank you. I'll be here next week doing the same thing again. So if you've got your questions, save them up and we can elaborate next week. I hope I see you all again next week. And many more, actually, because everybody's welcome. So happy breeding, happy growing, and good health to you all. Have a good week. Bye-bye. I'll give it 30 seconds a week again, just for this stuff to come through. Thank you. Thank you, everybody. Have a good week.